Proteins. Protein is a word which is actually made up of the word called proteose, which means very important, most important. So these are the biomolecules which are the primary biomolecules which are the building blocks of the living beings or of the life. They are the one which together constitutes or leads to the formation of the living organisms. That is why they are called building blocks. They are abundantly present in fish, meat, pulses, milk, cheese, etc. Chemically, these proteins are biopolymers. They are biopolymers of alpha amino acids. So the monomers of these biopolymers are alpha amino acids. So in fact, if we want to study and we want to understand the nature or the types of the proteins, we need to understand first the amino acids. So let us first understand amino acids. See, amino acids, as the name indicates, are those organic compounds which contains amino as well as carboxylic acid groups. Now, if the amino group is present on the alpha carbon with respect to the carboxylic acid, it's called alpha amino acid, like this one. So this is the main acid group we have. The carbon next to it is called alpha carbon, and since the NH2 group is present on this carbon, it is called alpha amino acid. Now, the amino acid, they can also have a side chain attached to this alpha carbon atom, which is denoted by R. Now, there are actually 26 amino acids, out of which 20 are alpha amino acids. So in the syllabus we have there is a list given of the 20 alpha amino acids. There is no need to go into the structures of those details, but you need to only know the number. So there are 20 alpha amino acids. Now all those 20 alpha amino acids differs from each other with respect to this R group. Rest of this structure of all these 20 alpha amino acids is the same. Now how these R differs, like the commonly used amino acids, common we see this, they are glycine. In case of glycine, this R group is H, and the symbol given to this amino acids is GLY. We use the first three alphabets of the name of these amino acids, and usually their tribal names are used, which indicates either their property or their source. Another next amino acid is alanine. So alanine contains R as the CH3 group, and we use its symbol ALA. Similarly, valine is the third amino acid which contains an isopropyl group. Isopropyl group indicated by VAL. So this is how we have the different types of the amino acids. Now let us go into the detail of the classification of amino acids. Amino acids are classified as essential amino acids and non-essential amino acids on the basis of that which of those are synthesized in our body. See, we need all those 20 amino acids in order to prepare different types of protein and enzymes in our body. Now our body can synthesize a few of these amino acids. The rest of the amino acids must be taken in our diet. Out of 26 total amino acids, 10 are the one which our body can synthesize. Rest 16 has to be taken from outside from that in our diet. So those out of those 16 which are taken in our diet, so they are very much required to be taken in our diet, are called essential amino acids. Now out of those 16, 6 are beta, that are not in the syllabus, so I'm talking about only those left out, 10 alpha amino acids. So those 10 alpha amino acids which are not synthesized in our body and are to be taken in our diets regularly are called essential amino acids. Like valine is one of the important essential amino acid. Non-essential, those amino acids which are synthesized in our body itself, so there is no need to take them in our diet, are called non-essential amino acids, because there is no need to take them regularly in our diet because our body is capable of synthesizing them. So like glycine and alanine, these simple amino acids are the ones which our body can synthesize, and they are called what? Non-essential amino acids. There is another type of classification of amino acids depending upon the number of amino or acid groups we have in the amino acid. Now that classification, according to that classification, now the amino acids can be neutral, they can be acidic, or they can be basic. Now, when the number of NH2 group are equal to the number of carboxylic group in the amino acid, it's called a neutral amino acid. Like we have, suppose, a glycine, which is an neutral amino acid. When the number of carboxylic groups are more than number of NH2 group, we call it acidic amino acid, because the more number of acid groups makes it acidic in nature. And when the number of NH2 group are more than number of acid groups, we call it a basic amino acid. So this is again one very important classification of amino acids, which decides their nature and the pH in which they survive. Then the properties. Let us understand the properties of amino acids. See, amino acids usually are colorless, crystalline substances which exist in the form of salts. In aqueous solution, whenever we put any amino acid, what important thing we notice is that they show self-neutralization. How? We have an acid group and a basic group, so they easily shows what? Self-neutralization. Now, due to this self-neutralization, the acid group gets changed to the what? Carboxylate ion, and NH2 group gets changed to the ammonium ion. Now, this particular type of ionic species, which contains both cationic as well as anionic charges or ends, is called Zwitter ion. So, what is Zwitter ion is an ion which contains both negative and positive charges, is called a Zwitter ion. Normally, we have an ion which is having either a plus or a minus charge. Whether it is, usually it is a cation or an anion. So, the ion which contains both of these is called Zwitter ion. Now, the Zwitter ion in aqueous medium therefore behaves like what? A neutral molecular species. It remains what? Neutral, doesn't migrate to any of the electrodes, and that is what we call it. So such point where this kind of change also takes place in a particular pH for amino acid is also called its isoelectronic point. Now, the Zwitter ion is a very important property of any amino acid, and we can easily identify the pH at which it changes to this Zwitter ion. Now, another which is important thing about the amino acids is that amino acids except the first amino acid, glycine, all are optically active, except because glycine is having this R group as H, so that this carbon remains, it is not, it cannot be asymmetric carbon atom because it contains the two same H. Now, so that is why except the glycine, all other amino acids are optically active. And what we do is we draw this structure like this, we draw it like this one, or we put it like this. It can be drawn like this, but that's not the correct Fisher projection, because if we have to draw the Fisher projection, the most oxidized carbon has to be on the top, and then if you are putting it like this, it can be NH2 here or H like here. So if we draw the exact Fisher projection, that's wrong then. Now, if the NH2 group is on the right hand side, we call it what? A D form. It's a relative configuration, as you remember I have discussed in the case of carbohydrates. And if the same NH2 group is on the left hand side, we call it what? L form. It's with respect to the glyceraldehyde molecular species. In case of amino acids, we assign DLL configurations with respect to the NH2 group. So this is how these are the properties of amino acids.